Welcome to this video. Now, yesterday, I think it was the day before, I rashly promised to do a little bit on immunity. So I'm going to do that now, but I'm not going to go too heavy into it. So it says anyone should understand this. And I was looking back in this, I wrote this book a few years ago, still around, physiology book, physiology notes. Now, what I wrote here was, I'm talking about immunity and the problem of infectious disease. And I've got influenza killed between 20 to 40 million people in 1919. Now, actually, it was 1918 to 1919, and it killed, we now know from new research that it killed more than that. It was 50 to 100 million people that died. And then I wrote this: it is likely that such a pandemic will strike us again. Now, the reason I wrote that was not because I thought of it and made it up, but because virologists had been saying this for as long as I'd been learning about things from virologists. So it's not new. We've known this is going to happen for some time. So let's hope we can get a bit of interesting background on the immune response. Nothing too heavy, as I said. Now, just a few simple terms here. Now, an antigen is anything that the body recognises as being foreign. An antigen. So the body recognises things that are part of the self, and things that are non-self are recognised as being foreign. So this is just a very simple diagram. It could be a virus or a bacterium. It's the same thing. It's an antigen. The body recognises as being foreign. And what you have on the surface of these foreign materials, the bacteria or the viruses, you have proteins. Now, these proteins are not human proteins. They are viral proteins or bacterial proteins, and the body recognises these as being foreign proteins, and we call these epitopes. So the epitopes are the proteins that the immune system recognises as being foreign. And that's what infection is. Infection is the presence of antigenic organisms, whether it's bacteria or virus or viral particles, in the tissues of the body. So all around about us, there's untold numbers of viruses, bacterial particles on the surface of the skin, even in our mouths, in our orifices. We have an untold number of viral and bacterial particles way up in the air, way down in the ground, everywhere. And yet inside the body, it's sterile. It's quite amazing. And if you have viruses or bacterial particles in the body, that is infection. So these antigens are recognised as being foreign because they have these foreign proteins on it. So if you're eating a particular sort of meat, you can tell what sort of meat it is from the taste of the proteins. Because like a pig will have pig proteins, and a cow will have cow proteins, and a virus will have virus proteins on its surface. It kind of makes sense. Now, what happens in the body is when the body is exposed to an antigen. So here's an antigen that the body is exposed to. Then there's particular types of white blood cells. This one's called a B lymphocyte, but it doesn't matter. There's these white blood cells. And these white blood cells detect the presence of the antigen. And there's a particular type of white blood cell that's most suited to detecting particular types of antigen. And then what this cell does is it thinks, oh, there's a problem here, there's an antigen here. I better recruit some help. So what it does is this cell starts dividing and produces lots and lots of copies of itself. Now, two cells or two organisms with the same genetic material are called clones. So we get this process called clonal expansion, and this cell makes, I don't know, millions, billions of copies of itself, millions, many billions of copies of itself. So we end up with lots of these cells here. They're like this cell, but they've been stimulated by the presence of the antigen. And this cell thinks there's an antigen present. This dark bit in the middle is just its nucleus, don't worry about that. And it makes lots of copies of itself. Now, what these cells do, these cells here are called plasma cells, and they produce huge numbers of immunoglobulins, which is just the big name for antibodies. So it makes huge numbers of antibodies. And it's these antibodies that will then get rid of the infection in this process called cellular immunity. So what happens, there's various things happen. These antibodies are really amazing things. They are actually like bodies. They've got like 2 arms. So they've got a body part and 2 arm parts. And what the arm parts do is they can grab the antigenic components that they don't want. They latch onto them and chemically bind onto them. So here we have an antibody here. This is not drawn to scale. On the real scale, the antibodies will be about this size maybe. So it's not drawn to scale. But that's the antibody there. And it's got a bit on the end here called a variable region. And that variable region is designed just to fit into the epitope of the antigen. And it binds onto it like that. So it's binding onto it, holding it. And that does a couple of things. When lots of antibodies bind onto an antigen, so there could be another one binding onto it there, look like that. Because they have this affinity, these high affinity receptors. Now, when the antibodies, these antibodies, bind onto the antigen, that will inactivate the antigen. It will stop it from working. So the antibodies are acting against the antigen because the antigens possess antibody generating molecules. They are antigens. And as we've seen, the arms can move. So we get these antibodies binding to the antigens. The antigens are the viruses or the bacteria, inactivating them, and it also clumps them together like this. So here we see some antibodies have clumped together, lots of antigens. So that's an antigen, that's an antigen, that's an antigen, that's an antigen, that's an antigen. And now they're all clumped together by these antibodies. Now that's good because that's going to inactivate the antigens. But it also means we've got a big clump of them now. And then big specialised cells called monocytes are in the tissues, macrophages, will come along and they will actually physically engulf. They will put arms round about these things like this. And they will physically engulf them. They'll take them into the cell. So you end up with this clump inside the cell like that. So the cell actually eats them. And then specialised units within the immune cell, in this case a macrophage or a monocyte, will move towards this clump of antibodies and antigens and will physically digest them. They're called lysosomes. And they will release an enzyme called lysozyme that will physically digest these and turn them into fats and proteins, just as if they're a piece of meat being digested. And of course that will completely inactivate them. So it depends on these antibody things. They're quite amazing antibodies. So they've got a hinge bit and they move, and they've got these variable regions that clamp onto the antigen. So this is important to know in terms of the COVID-19 because we have these different sorts of tests. We have the antigen test that tests for the presence of the antigen. That is for the presence of the virus. And that tells you if someone has the infection at the time. It tells you if they've got the infection, and it also tells you that they're likely to be infectious because you are detecting the presence of the antigen, the presence of the virus in this case. So the COVID-19 virus is the antigen. The antibody is what your body makes in response to that to eliminate the virus, to inactivate it by clumping onto it, and to clump it together so it can be eaten by these immune eating cells called phagocytes that eat. Phag means to eat, cyte, cells. Eating cells, big eating cells, phagocytes. But the point is that the antibodies are left in the blood after the infection is resolved in case the infection comes back again. Now, I know there's debate going on at the moment as to how much immunity this confers, but with most infections, you're going to be immune to it after you've had it because you have the antibodies. So as we test for the antibodies, we can see who has had the infection in the past, therefore who is likely to be immune to the infection. And it will also tell us, when we test lots of people in the community, how many people have had the infection. So that means we can work out what the case fatality rate is and the complication rate is. So we're waiting for these antibody tests in most areas of the world. So that's why that's important to understand, because we have the antigen and the antibody tests, and we need both in massive numbers, as we've said repeatedly, because that is the exit strategy from the lockdown that we are currently in at the moment. So that was just a bit of immunity. I'll post some more below on the descriptions. If you want to read it, then do feel free.